Picture this for a second. Your body is like a massive high school, thousands of students moving through the halls. Most of them just want to learn and do their thing. But a few get a bit too excited. They start fights, pull fire alarms, run into classrooms where they don't belong. If nobody is there to calm them down, to say, hey, not now, not there, the whole school turns into chaos. Now here's the scary part. Your immune system works exactly like that school. And this story, noticed and carefully unraveled by Mary Brunkow, Fred Ramsdell and colleagues and published in Nature Genetics, is about what happens when the principal of that school simply doesn't exist in 0101_68. Why you should care? Yes, really. Because autoimmune diseases, allergies, severe childhood immune disorders, and even some modern cancer treatments all trace back to one core question. How does the immune system know when to stop? This paper doesn't scream, it whispers. But what it whispers ended up changing medicine forever. The mouse that couldn't say enough, let's start with a mouse. A tiny one, scruffy fur, dies before it's even a month old. Researchers had been watching this scurfy mouse for years. They noticed something deeply unsettling. Its immune system wasn't weak. It was too strong. Way too strong. The mouse's T cells, immune cells meant to protect, multiplied like crazy. They flooded organs, skin, liver, lungs, cytokines. The immune system's chemical megaphones were blasting nonstop. Inflammation everywhere, no off switch. This wasn't infection. This was the body attacking itself. Brunkow and Ramsdell noticed something important here. This looked eerily similar to other mice missing key immune breaks like CTLA4 or TGF-beta. Different genes, same chaos. That told them something profound. The immune system doesn't just need weapons, it needs restraint. Hunting the missing break. So they went gene hunting. Back then, this was slow, painful work. No AI, no instant genome browsers. They narrowed the problem down to a tiny region on the X chromosome, about 500,000 DNA letters long. Imagine trying to find one typo in an entire library by reading every book manually. Inside that region, they found about 20 possible genes. 19 were innocent. One wasn't. They discovered a gene encoding a protein from the forkhead/winged-helix family, proteins known for controlling other genes. This one was new, never described before, highly conserved in humans. They named the gene FOXP3, and the protein it makes? Scurfin. Already that name tells you something. This protein was missing in the scurfy mouse. The smallest mistake with the biggest consequences. Here's where it gets wild. The scurfy mouse didn't lose the whole gene. It didn't delete FOXP3. Instead, it had a 2-letter insertion in its DNA. Just 2 extra chemical letters. That tiny mistake caused a frame shift, like adding 1 extra letter to a sentence and suddenly every word afterward becomes gibberish. The result? A broken protein missing its critical DNA binding domain. The part that tells other genes what to do? Same gene, same instructions, but now unreadable. And without that working protein, the immune system had no chill. Proof, not guessing. Good science doesn't stop at "this looks suspicious." So the researchers asked the hardest question possible. If we give the mouse a working copy of FOXP3, does it live? They created transgenic mice, scurfy mice with an extra healthy FOXP3 gene added back in. And something incredible happened. The mice survived. No massive immune attack. Normal lymph nodes, normal organs. Same genetics otherwise. This wasn't correlation anymore, this was causation. FOXP3 wasn't associated with immune control. It was essential. What FOXP3 actually does. Let's simplify. Your immune system has gas pedals, cells that attack, multiply, and destroy. FOXP3 is part of the brake system. Specifically, it controls a group of cells we now call regulatory T cells, the peacekeepers. Their job is not to fight, but to prevent fights from happening unnecessarily. Without FOX3, T cells don't calm down. Immune responses don't shut off. The body attacks itself relentlessly with FOX3. Immune responses stay proportional. Damage is limited. Balance is maintained. Think of FOX3 as the group chat admin who can mute everyone when things get out of hand. Where FOX3 shows up and where it doesn't. Brunkav's team looked across the body. FOXP3 wasn't everywhere. It wasn't in the liver, it wasn't in the brain. It showed up mostly in thymus spleen-specific CD4 T cells. That specificity matters. It told researchers FOXP3 wasn't a general-purpose gene. It was a precision tool for immune regulation. Humans were next, and the implications were huge. Around the same time, other researchers noticed something chilling. Children with a rare, deadly condition, severe diarrhea, diabetes, endocrine failure, immune chaos, had mutations in the human FOXP3 gene. Different species, same gene, same disaster. This mouse wasn't just a mouse anymore, it was a mirror. A real-world example you can imagine. Imagine a 6-year-old child. They're born healthy. Then within months, food causes inflammation. Skin erupts in rashes. The immune system attacks the pancreas. Hormones collapse. Doctors treat symptoms. Steroids, immunosuppressants, Band-Aids. But the cause? A single broken gene that was supposed to teach the immune system when to stop. Understanding FOX3 gave medicine a target, a reason, a direction. Today, this knowledge shapes autoimmune disease research, transplant medicine, cancer, immunotherapy, allergy treatment strategies. All because someone cared about a scruffy mouse. The deeper message, and this is important, this paper quietly teaches a bigger lesson. Strength without control is destruction. The immune system isn't powerful because it attacks. It's powerful because it knows when not to. FOXP3 isn't about fighting disease harder, it's about fighting smarter. Let's repeat what matters, because repetition is how it sticks. The immune system needs breaks, not just weapons. FOXP3 is a master regulator of immune balance. Tiny genetic changes can cause massive systemic failure. Mouse models can reveal human disease mechanisms. Autoimmune diseases aren't random, they're failures of regulation. Say it another way, your immune system doesn't fail because it's weak. It fails because it forgets restraint. Where this leaves us, Brunkow, Ramsdell, and their colleagues didn't just identify a gene. They revealed a principle. That health depends on balance. That control matters more than force. That sometimes the most important molecule is the one telling everyone else to slow down. And the main outcome? FOXP3 is essential for immune homeostasis. Without it, the immune system turns on itself. And with it, balance can be restored. That idea still echoes through medicine today.